Cell structure, part 3. Here we have a good example of a generalized animal cell. If we start at the center, upper of part of this image, what we'll see is that unique structure that we find in all cells that we call eukaryotic cells. So this is an idealized, generalized animal cell with a well-defined nucleus. The nucleus is the location of hereditary material in the form of chromatin, which is a combination of DNA and proteins. The chromatin, for the most part, is arranged in a very lacy arrangement, very loosely held together inside the nucleus as it's being utilized or used for the everyday function of the cell. The chromatin actually is organized in various regions, so this is not a haphazard arrangement inside the nucleus in terms of the location of the chromatin, but it is where all of the instructional information on how the cell is to function is warehoused. In addition to the chromatin inside the nucleus, we also would find a nucleolus. The nucleolus is a specialized structure for the formation of what's called ribosomal RNA for the function and structure of ribosomes. The nucleus in a eukaryotic cell is a very well-defined structure that has a very specific outer covering called the nuclear envelope, or it can also be called nuclear membrane. The nuclear membrane or envelope will only allow certain types of materials to pass inside and out to the rest of the cell from the nucleus. So the membrane itself for the nucleus is a highly organized, very specific structure with pores, what we call nuclear pores, and that's what's illustrated in these little gold-colored structures surrounding the surface of the nucleus, are the nuclear pores that will only allow certain materials inside and outside of the eukaryotic cell. As we go through the rest of the slides, we're going to describe and discuss some of the additional structures that we'd expect to see inside a typical animal cell. Assorted types of folded membranes. We call this an endomembrane structure. We'd expect to see in a eukaryotic cell, starting with what we call endoplasmic reticulum. Endoplasmic reticulum is going to be a continuous folding of membrane structures that's intimately attached to the nuclear envelope. We have two types of endoplasmic reticulum, rough and smooth. The appearance of the rough is because embedded on the surface of this endoplasmic reticulum are the ribosomes. That makes it appear to be rough. The smooth endoplasmic reticulum has either no ribosomes or maybe just a few. But usually when we think of smooth endoplasmic reticulum, or ER, so SER is the abbreviation for smooth endoplasmic reticulum, we actually generally define those as having no ribosomes. The RER, or rough endoplasmic reticulum, we always presume that telling us that the ribosomes are attached. Close to and associated with the function of the endoplasmic reticulum is what we call the Golgi apparatus. The Golgi apparatus is a sort of a finishing school for all of the materials made inside of the cell, or at least most of the materials. We also would see these orangey structures that are labeled mitochondria. Those are like little powerhouse factories inside of the cell. We might also see centrioles, illustrated in the golden yellow color, sort of at the center of this cell. We'd also might find lysosomes and peroxisomes inside of the cytoplasm, or floating in the cytoplasm of a eukaryotic cell. In addition to these internal structures, collectively called organelles, We might also see other additional structures like cytoskeletal material in the form of different kinds of filaments, and even changes in the surface of the cell, such as the formation of microvilli, depending on the type of cell we're working with. The outermost barrier for every cell is going to be a phospholipid bilayer called the cytoplasmic membrane. Now, most eukaryotic cells will have a phospholipid bilayer. For prokaryotic cells, there are some exceptions where there's no bilayer, but instead a monolayer of phospholipids. But that's kind of the exception to the general rule. In general, cytoplasmic membranes have 2 layers of phospholipids with proteins attached, or what we call embedded, in that bilayer, which is illustrated here in this cartoon drawing. The cytoplasm is the region between the plasma membrane and the nuclear envelope and consists of the gel-like material called cytoplasm, or cytosol, plus the cytoskeleton. The majority of the cytoplasm is water, and the rest is suspended chemicals and dissolved chemicals. The nucleus, as we've already said, is the location of the genetic material in the form of chromatin, which is a DNA protein structure, and the nucleolus, which is the location of ribosomal RNA formation and the construction of ribosomes. The endoplasmic reticulum and the nuclear envelope are intimately associated with one another. So there's essentially a causeway or a pathway between the two. The little cartoon drawing in the upper right of this image shows you the detailed structure of the nuclear pore complex. That these are special channels or pathways through which materials can enter into or exit the nucleus. So we have very tightly controlled transport between the nucleus, internal function, and the rest of the cell for a eukaryotic cell. For the nucleolus, we're going to find again that specialized structure in the nucleus of a eukaryotic cell where ribosomes are assembled from ribosomal RNA and proteins. We're going to find that ribosomes are called protein factories, and we'll talk about those toward the end of the semester when we talk about how these different components that we find in a generalized cell can function. For the mitochondria, those are the little powerhouses of a cell. Mitochondria are very unique because they can function somewhat independently of the rest of the cell. Mitochondria undergo their own type of replication, or you could think of it as division, making copies of themselves, which is illustrated in the photo, which is actually fairly rare to find in any kind of resource. The photo on the lower left is an image of mitochondria undergoing division, making a copy of itself. Mitochondria are usually illustrated in most textbooks and in most photos as having this sort of hot dog or sausage shape, but for certain types of cells, mitochondria can have very different types of shapes. But the general configuration and the general common feature for all mitochondria is that it's a 2-membrane structure. It has a biological membrane or a phospholipid layer. It's called an inner membrane, and then it has a bit of a space between the inner membrane and what's called an outer membrane. The inner membrane has infoldings called cristae, and inside that inner membrane is a gel-like material called the matrix. We'll talk more about the function of mitochondria as we get through the chapter on metabolism, but keep in mind the mitochondria is the powerhouse or the location for the formation of chemicals that have potential energy to do work and that chemical is called ATP. Mitochondria are tiny little organelles that you could see inside of a cell with a very specialized stain. And in fact, most of the organelles, with the exception of the nucleus, most organelles in a eukaryotic cell cannot be viewed with a light microscope. Mitochondria and also Golgi apparatus are the exception, with special stains, as illustrated in this photo. All of the little oval-shaped, pale gray items, the tiny ones that the arrows are pointing toward, you can see for most of these cells in this photo, there are hundreds of these little structures inside of the cell. Those are the mitochondria. So it's a unique type of image and a unique type of photo because most of the time when you look at a eukaryotic cell, you cannot see the mitochondria unless you use a special stain to reveal their presence. Another structure that you'd find in a eukaryotic cell is a peroxisome. A peroxisome is a particular location for what's called the breakdown of fatty acids and amino acids. Occasionally, a cell needs building blocks. And to get those building blocks, it has to take a complex structure and break it down into smaller parts. And that's what peroxisomes are good at doing. Peroxisomes might also be a location where toxins or poisons can be torn apart. Not all types of cells have peroxisomes, but when they are present, that is one of the functions that can occur. And notice, for a peroxisome, it too has a type of membrane structure.